Today I wanted to share with you some of the work that I do in terms of gene-environment interactions, but I also I have gone into the literature and borrowed generously from the work of others to try to give you information about where we are in terms of gene-environment interactions. For me, I think the way to look at it is really, it's a concept of co-evolution. Right now, I'm extremely interested from a scientific point of view, the whole notion of what are the forces that shaped our genome, and what are the footprints that those forces have left in our genome in terms of our health and disease susceptibility, and perhaps just as equally important is who we are from an ancestry position, and how we are related, to each other. So, from my own lab and from my training from this School of Public Health, it's really, I try to model my work in the context of gene-environment interactions, and this is just one, model that I tend to always try to refer to. But this is for type 2 diabetes. I think you can draw the same thing for almost all complex diseases, which is basically you have a genetic background and you have some environmental exposures in terms of lifestyle characteristics, intrauterine microbiome environment, and how all of those present themselves in terms of basic physiology, intermediates, and then drive you towards a specific outcome like diabetes. It could be any other outcome. So this is just again an example of how I try to model this in my own lab. So sort of starting from the environment, what are some of the characteristics in the environment that drives us towards different directions in terms of health? And I think it's important when we discuss this to put it in the context of disease etiology and also disparity. These are two circles, and these circles, they overlap, but they are not the same thing. Okay, so if you're trying to understand etiology, that again, you basically answering some fundamental biological questions. And but if you're trying to understand disparity and shed light on disparity, the underlying physiology can be the same across different populations, but it's the force of exposure that becomes different. So I think that I'll try to show that in my talk, today. So there's the whole issue of lifestyle, and the issue of urbanization. Actually, those yellow buses, that's somewhere in Lagos, Nigeria. So it can be quite crowded. Access to care, again, the whole issue of the rate of incarceration in the United States, for example, and tax policies. Those are just as important as smoking, in terms of putting people towards a poorer health condition. So these are some of the environmental factors that I do consider in my work. And you look at something like obesity and diabetes, it seems to be we want to call it brothers and sisters in crime. So the rate of obesity is tremendously going up, and if you overlay the rate of diabetes, you see that obesity is a major driver of people towards diabetes. But the point I want to make here is that, are we evolutionarily set up, to go in this direction? Because one of the questions I keep asking myself is that, why is it that your body almost always wants more food? Why is it that you eat and then you walk past that cookie and you smell that thing and you just want to take it again? So why is that? So I think there's some evolutionary context to the need for maybe having survived certain environments and the ability to store fat and things like that. And hopefully, I can provide some examples of that. But those are all working hypotheses. And how true these are, I think, requires more vigorous research. But clearly, we were not designed to sit in one spot. And I don't think we were designed to consume as much calories as we do today. I'm just as guilty as the next person, in a sense. So how do we bring all of these things together? One of the things that we know in terms of the etiology of type 2 diabetes, for example, is that genes are very important but not sufficient. You can have a very strong family history and still not develop diabetes, or at least delay it, for a much longer period of time. But there's no doubt that there's a very strong family history and a clustering of diabetes. For example, identical twins is 75%. That if one twin is diabetic, that the other one will be. And we have also identified several candidate genes. Over 400 loci so far have been identified for type 2 diabetes. One of the most powerful obesity gene that has implications for diabetes is the FTO gene. But the effect of that gene actually depends on the environment. Okay? And so again, individuals who carry, the high-risk form of FTO really do have, can modify that risk by exercising and also watching what the amount of calories that they consume. So it's clearly an opportunity for gene-environment interactions. So we also know that lifestyle factors alone is definitely not enough. Although I showed you a map earlier where you see a very good overlap between obesity and diabetes, but majority of obese people are never going to be diabetic. Okay, so we need to have that also at the back of our mind. So 30% of overweight people have diabetes, but 85% of diabetics are overweight. So you can see that there's a very strong relationship, but it's not a one-to-one correlation. Okay? And perhaps just as important is that there are very, very many lean people who have diabetes, and they've never been fat in their life. Okay? So we also need to be very conscious of that. So this is the thing about the gene-environment interaction. So if you are obese and you have a strong family history, you can see how dramatic your risk is in terms of developing diabetes. So again, the gene and the environment coming together. In this case, we are saying obesity is environment because it's highly driven by lifestyle. But I'm also going to show you information now that says that obesity itself may also be under genetic influence. And I think I discussed this study earlier with Tony. I don't know if Tony's here. Yeah, he's over here from New Zealand. And there are some caveats here in some of the things I'm going to talk about. We discussed this earlier in terms of local adaptation and maybe some of the concepts I want to raise here. And that's the whole notion, is that there might be forces in our genetic background that may also be increasing our how heavy we get, even under exposure to similar energy consumptions. The Samoans, I think, are one of the biggest people in the world, I think, on average. So 80% of Samoans are indeed overweight or obese. So it was not surprising when a study using the whole genome genome-wide association study approach in about 3,000 Samoans, and found this CREB3 regulatory factor that strongly influences BMI, and they picked up this angenin substitution here in the context of BMI, and identified a variant that may be under a very, very strong selection pressure with about almost 30%, and very rare in other populations around the world. So again, sort of providing evidence for why these individuals may indeed be at much higher rate of obesity. So functional studies show that this variance decreases energy use and increases adipose fat storage. So this, again, you can see how this can lead to somebody, being very, very heavy in sense. So part of the hypothesis that were pushed by this investigator is the whole notion of the tryptogen, tryptogen hypothesis, which basically says that because as humans, we were hunter-gatherers some years ago, 60,000 or so years ago in different African communities, and that we needed to be very have the very strong ability to conserve energy and store fat. And therefore, if you have such genetic background and you're now put in an environment where food is, in essence, where you can actually get pizza and all those kind of things, that you may indeed be at higher risk of developing obesity. But again, these are things that still needs to be tested and understood. Is this true? Is that selection pressure strong enough and all that? But this is again clear evidence, I think, that obesity itself may be under genetic influence, despite a very strong environmental influence in terms of lifestyle. So another example here is the Neanderthal introgression. For me, as somebody who runs a genomic lab, one of the really exciting things that I see is our ability to understand biology for the very first time in a way that we couldn't. We couldn't do it without this kind of genomic approaches, because we know that we all started somewhere in Africa. Then some of us were very restless, or for whatever reason, we decided that we wanted to venture to different parts of the world. And interestingly also is that when we do venture, carrying a subset of the genetic variation that existed in Africa then, there was introgression with Neanderthal, other hominids in this outside of Africa, Europe and Asia. And there is evidence in our genome now that we indeed do have components from Neanderthal in genomes of individuals who migrated out of Africa. About 2 to 4% of the genetic materials of those individuals are indeed from Neanderthal. So the question is, what is the implication of this for human traits and diseases? So if you ask that question, there was a very, very good study that was done recently that looked at diabetes among Mexican and other Latin American populations, and they identified 2 solute carriers that were very, very highly significant with a very reasonable odd ratio of about 1.3. And it was actually quite interesting. For me, what makes this study really fascinating is the fact that this particular haplotype under which the background disease is operating is rare in other populations, but quite common in individuals who self-identify as Mexicans, and a little of about 10% among Asians. Now, when David Reich at Harvard and his group looked at the background, genetic background for these variants, it turned out that by looking at archaic genome sequences, that this particular variant may have been introduced into the gene pool by the admixture with Neanderthal, which is absolutely fascinating. Again, sort of giving us some information and the power of genomics. I don't see how we could have gotten at this from biochemistry or from type work. It just doesn't. We don't have the power, to be able to understand this. So again, how important this is, I know some people are trying to create, actually use this as a drug target. How successful it's going to be, I think, is still a question under investigation. But again, it really does show that genomics can indeed be quite powerful in understanding disease, even as complex as type 2 diabetes. So this is a recent example from my lab in terms of type 2 diabetes. This paper is under review now. We've gone through the second review in Nature Communications. Long time ago, Francis Collins was a missionary in Nigeria, and while he was there as an internist, he actually treated quite a few diabetics. He was quite surprised, to see quite a few diabetics, and that stayed with him. So when he came back to the United States and started heading the Genome Institute at the NIH, in collaboration with Georgia Dunstan, who was a professor at Howard University, they actually say, how can we study diabetes in African countries with some idea of how we can inform what we see in African Americans? So they invited me to be to be the PI of this study, which we did, and we've been doing now since, I want to say since 2003, I think. So this study has been going on, and in Ghana, Nigeria, and Kenya. And we've enrolled over 8,000 individuals from these various populations. But recently we did a GWAS on over 5,000, and we picked up, these are known diabetes gene, but we picked up this new variant, new gene here, ZRAMB3, which has not been reported before in any population. And the reason we think is that the variants that we pick up here is African specific. It's monomorphic in other populations. So we were quite, surprised, and but at the same time, very pleased. But when we started to search the literature to see what does this gene do, there was literally no information on it. We saw some expression work which encouraged us that it's actually expressed in the pancreas, in the liver, and some other, tissue. But we were quite surprised that there's not a whole lot known about it. So we started doing some zebrafish work, and we started saying, what happens if we knock down this gene in zebrafish, at least the ontology, in zebrafish. And to our surprise and amazement, it actually led to reduced beta cell number, as you can see, here, using both CRISPR and morpholinum. So we were quite happy that we were able to demonstrate this, and then we wanted to know what is the consequence of knocking down and having this fewer beta cell mass, compared to controls. And so we use the mammalian cell lines to actually, again, this is zebrafish, mammalian cell line to try to understand what happens if we silence this gene, and it turns out that there is massive reduction in ability to respond to high glucose load. So again, really encouraging us to push ahead. So we went ahead and we crossed the F1 generation to get it F2 homozygotes, and we were still able to see the difference in beta cell mass, and we were able to also demonstrate, because one of the things you want to do is to also demonstrate that it is not as a result of damaging the whole animal. So we're able to show that glucagon was not affected, again, by this manipulation that we are doing, at least in the F2 generation of this. So we'll send this paper back now, and hopefully 2 of the 3 reviewers have said yes, but the zebrafish expert, I think, is putting us to more task. So hopefully we've been able to convince the person with this new data, and we hope to hear any day now. Again, this is a very good example of why we have to apply genomics to different parts of the world, because this particular gene, if we study it well enough, it may indeed become beneficial to all human population, but we couldn't have gotten there if we have not studied an African population where it gave us more power to be able to find the gene in the first place. Okay? Just like the example with the Mexicans, I mean. So again, trying to show to you that obesity itself is still genetic. This is another finding in terms of African populations. We picked up this variant in sigma 4D. Individuals, although it's about 1% prevalent, but individuals who carry this particular variant are about 6 pounds heavier. Think about that for a minute. At the population level, that is quite significant. And again, as we study this in our lab, we hope to share more light on how this actually influences obesity level and how it may indeed be relevant even for metabolic disorders like diabetes. Again, it would not have been picked up if we have not been studying African population. You will hear that from me over and over again because I really want that message to get through, that genomics is a global enterprise that we need to do. I'm going to skip through this. Again, this is just the same kind of study, but for metabolic disorders, what we call metabolic syndrome, where we picked, again, identified some African-specific variants with a very strong odd ratio in terms of risk for developing metabolic syndromes. Now, this is one study I want to spend some time on. I'm sure some of you here either are participating or already part of the CHARGE consortium, and that's where this data is coming from. This is a paper that led by Amy Bentley in my lab. It's just been accepted in Nature Genetics, where we studied, again, a very large number of individuals, close to 400,000 from multiple ancestry background. But the question here was, is there a gene-environment interaction in terms of the lipid profile with gene and smoking? So, we tested multiple models, but what I want to discuss with you is the interaction model. Again, what we call the 1 degree of freedom interaction. Basically, this shows very clearly that there are variants or genes that we can pick up from the genome by taking into account environmental factors. And if you do not take them into account in terms of interaction, you will not find these particular genes. And it is not sufficient to adjust for those environmental variables. So for example, if you had a standard regression model or logistic model where you are controlling for smoking, you will not pick up these variants. You actually need to test the interactions. Okay? So that's sort of the key message here. And the other thing also is that most of these findings were in African ancestry populations, 9 out of 13 of them. And we thought that most of them could have been explained by just the fact that there's differences in allele frequency. But that wasn't the case. There were some that were due to differences in allele frequency, but there were others that, despite higher frequency in other ancestral populations, we did not see those relationships, but only in African ancestry populations. As you will see here, these are the African ancestry populations, and non-Africans, there's really no association whatsoever. So again, it's pointing at the importance of interaction. Now, this is highly speculative, but we went ahead and made this speculation anyway. And that is, why is it that we are seeing such strong interaction between genes and smoking in the context of lipids in African ancestry individuals, in this case, African Americans? And part of our speculation was that when we looked in the literature, there is some data that says African Americans at a higher proportion tend to prefer menthol cigarettes. Okay? And there's also evidence in the literature that says individuals who smoke menthol cigarettes tend to inhale more because it's soothing. Okay? Now, the reason I say it's highly speculative is that this particular gene here actually has implication for taste receptors. And we were wondering whether our ability to capture this particular SNP in this gene, could it be related to that preference? that we see. And I'm telling you, we are going to pursue this at the experimental level and try to show if this is indeed true. So for now, take it as highly speculative and put a very big grain of salt on that speculation. But hopefully in the next year or coming years, we'll be able to show this using functional assays if it is true. Now, this is a story I want to spend some time on and really show to you why it is important how we label people. I think we were having that discussion earlier. Who is African American? This study will really shed some light on that for us to be very, very careful. There have been other studies that have done some work similar to this. But for this, I have always been amazed and surprised by the fact that despite a diet that we say otherwise, African Americans have one of the best lipid profile in the United States compared to other ancestry groups. But if you look at the diet, you will say it shouldn't be so. So for me, it's sort of saying, is there something else that is driving this? So the first thing that we did in my lab was really to say, what happens if you plot lipid levels against the degree of African ancestry? Okay, so you will see here that triglycerides go down as the proportion of African ancestry increases. HDL goes up as the proportion of African ancestry increases, which for me was a clear motivation to go and look more specifically at the genes. So in my lab, we looked at, you're going to see a Bentley name whenever I say lipids, because in my lab we have people that drive different metabolic diseases. Amy is mostly lipids. So we sequenced 5 genes that is already known to influence lipids. And we basically asked the question, can they help us to explain these things that we see? We were actually quite surprised for the lipase gene, the LPL gene. We saw this variance that indeed influenced the rate of triglycerides and HDL, but in a very specific way. Okay? So if an African American carried this variant under an African background, under a European background, their lipid profile looks exactly like that of a European. But if they carry the same variant under an African background, they look like West Africans. Okay. Which shows very clearly that the lipid profile that we see in African Americans is being heavily influenced by the ancestral background in which this thing is operating, and that you cannot look at African American as one uniform group if you are indeed trying to explain phenomenon like this. And there are multiple examples of this in a sense. So I always say that for me, the only way I can describe who an African American is the descendant of the Middle Passage. Outside of that, all bets are off, because there are multiple ancestral backgrounds, there are multiple input into the gene pool and other factors that have driven that. So that's why I think this is really important for us to be conscious of this. This is another example, and I know people are working on this in this, and I met with your group earlier, working on the APOL1. But this, again, is another example of the adaptive processes and how that has shaped our genome. I'm sure quite a few people here have heard the APOL1 story. But before we get to the APOL1 story, it's really to understand the huge disparity that exists in terms of kidney failure in the United States and globally, really, especially in African ancestry populations. African Americans are 13% of the US population, but accounts for 32% of kidney failure in the US. Okay? and they're 4 times more likely to develop kidney failures, in a sense. So the hypothesis was that if this is indeed genetic, then if you use admixture mapping, that they should track the West African ancestry. Okay, that makes sense. If it is genetics, and since you've compared them to European and they still have a higher rate, then the other contributor to the gene pool is West African population. And this was formally tested. And in 2008, these 2 papers were published together in Nature Genetics that picked up this variant, on chromosome 22, which was originally thought to be MYH9 locus, which again was quite logical in terms of kidney disease. But on sequencing and using sequence data, it turned out that it's actually the culprit is 2 variants in the APOL1 gene that had an odd ratio of almost 11. But what was really outstanding about it is the fact that the frequency was quite different in different populations. Among the Yoruba, it was about 40%, among African Americans, about 20%, and absent in Europeans and Asians for the most part. So again, evolutionarily, as a geneticist, you want to say, okay, why would something that is bad rise to such high frequency? Okay, so the story of the malaria and sickle cell and all those things sort of start to come to mind. And it turned out that the APOL1 variant is actually quite protective against a deadly African sibling sickness in terms of trypanosomiasis exposure, because it lyses trypanosomes. That beginning to make sense. And that so when you started to look at it and look at existing data, especially from Friedman, in terms of Wake Forest, it became quite clear that this is maybe driving the health disparity in a very strong way that we see in terms of kidney failure. So about 1 in 5 people with 2 copies of the APOL1 risk variant will develop kidney disease. And the risk variants account for 70% of non-diabetic kidney failure in African Americans, and about 4 in 10 who are on dialysis as a result of this gene. Again, that is extremely powerful in terms of explaining the disparity that we see in terms of kidney disease. So there is an additional message here. So if you look at the global distribution of this variant, you see that it is practically everywhere in a sense. So you have to ask yourself why. We just say it's African specific, okay, to a very large extent. So the question becomes, who to screen? Who do you screen if you were actually going to implement this in terms of a public health contest? Individuals who do not self-identify as Africans or of African ancestry may indeed carry this variant because of admixture. They may indeed have an African background that they are not aware of, and they may actually be putting them at risk. But if we were making a public health recommendation and we were using phenotypic descriptions, then, or self-identity, we may be reaching the wrong public health decisions. So it is critically important that we understand that individuals who we may not consider to have this background may indeed have it as a result of admixture. Okay. The other thing that is highly instructive right now is the whole issue of microbiome. My lab is just beginning to put a little toe into the water of microbiome. But it is amazing to me, as a scientist and also somebody who's interested in history, it's very interesting to me as to how science develops. I know definitely when I was in the School of Public Health, I don't even know we mentioned the word microbiome once. Okay? But now it is absolutely clear that the microbiome that we all carry is critically important to our health. Even our ability to respond to some drugs is under the influence of the type of microbes that exist in our guts or different tissues. So, the reason I'm bringing this here, this is the School of Public Health, and I think the more we embrace this kind of thinking and this kind of science, I think it's going to create a competitive advantage if you are able to set up your study to begin to bring this kind of questions into your design and the way you want to pursue your science. So, what are some of the evidence that the microbiome is indeed very, very important in terms of our health, and how are they influenced? We know what you eat is very important to your microbiome. Even your stress level is important. But look at what, just by Charles Rotimi leaving Nigeria and coming to the United States, I've radically changed my microbiome constitution. Although I've been here too long now that I don't think I even have the Nigerian part anymore. But it is very, very clear that as people migrate from an environment that has created a very diverse microbiome into an environment that is less so, that you do lose some critical structures and diversity in your microbiome in such a way that it may actually be putting you at increased risk for metabolic disorders. So, talk about gene-environment interactions, and so I think we really need to pay attention to this, and I think we are just beginning to scratch the surface in terms of the microbiome work. So in my lab right now, type 2 diabetes study that is going on in Nigeria, Ghana, Kenya, we have now included collecting stool samples, okay, so that we can indeed properly adjust our gene-environment interaction models to include information from the microbiome. Because we now see some very clear evidence. This to me was convincing that if you take microbiome, from individuals who are, from mice that are obese, and you inject them into germ-free mice, and you can actually make them obese. And if you do the opposite, from lean and they stay lean, to me that was highly, highly convincing about the influence of microbiome, and to the point where I'm now actually including it in my own study design to try to understand how microbiome influences our health. And for diabetes, it's actually quite, again, important in terms of treatment, because the major #1 drug for type 2 diabetes, metformin, and it is now clear that metformin action is actually in the gut and is influenced heavily by the microbiome. So apart from actually your risk of the disease, it's also influencing the type of treatment or how your drugs work. So again, very, very important. I'm winding down now. I'm not sure how much time I have left, but I wanted to touch on where you live is critical to your health. I think the concept is location, location, real estate. I think we should be saying the same thing for health. Location, location, location. So what do I mean by that? If we just do rural-urban comparison for a minute, about 50 million Americans live, in what we classify as rural environment. But we also know that rural people or residents tend to be older and sicker, and they have more preventable deaths, and they have a higher rate of cigarette smoking, high blood pressure, obesity, and poverty, and lower leisure time physical activity, and lower use of seat belts, less access to health care, and less likely to have insurance. For me, this is a scandal that is not adequately recognized in public health. The rate of suicide is talked about, but I think it should be a national alarm, just like the rate of incarceration. Why would rural people be committing more suicide, than the urban centers? To me, I think there's something there that needs to be really addressed, and I don't think it's talked about enough. And it's the same thing at the international level. If you look at a place like Nigeria, about 80% of rural populations are almost completely excluded from the healthcare services. But the story of urban and rural is not as clear in some contexts. If you look at something like cardiovascular or blood pressure, the rate of hypertension, certain things in the rural environment in developing countries are still protective, but it's not true in developed countries. So the rural environment in Africa, for example, is very different from the rural environment in the US. Cardiovascular risk factors, stroke, and all of that in the rural US population is much higher than the urban environment, but the opposite is true in Nigeria. The rural dwellers in Nigeria and in a lot of African countries are actually healthier cardiovascularly than the urban dwellers because there's less obesity and they are more physically active and their diet is still not as high calorie dense, although all of those are changing, with time. So there is a little bit of a caution there. But overall, the rural environment really do put people at increased risk and less access to health. This is a clear demonstration of location, location, location. Again, I think the last time I was here, I showed this slide, and I highly recommend this book. It's not a difficult book to read, but it's a book that will challenge your emotions, and it will challenge your concept of how health is really managed in the United States. It's a physician, one physician experience, documenting his practice over years in Chicago. And he wrote this book that I read and I couldn't put it down, despite the fact that in some areas I was emotionally challenged. And he showed very clearly that individuals who live just within a football field or so apart can have such radical differences in life expectancy. Okay? This is Hyde Park area in terms of Chicago, if you know that area. Very close to Washington, Park. This is predominantly African Americans. This is mixed. Some of my friends live here. But you see the difference in life expectancy and, of course, income. Again, the same thing here in terms of the loop, in Chicago. But what this book really shows is that how people were making decisions about whether to pay their rent or pay for drugs. Whether to step out, whether even they have shoes, because you know how cold Chicago can get, so that they can go to work, and how all of that environment put them at risk. Even the advice that physicians give to individuals, just because of the neighborhoods they were coming from, were very, very influential to their long-term survival and health. So, again, location, 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 and health disparity. And I'm going to end my talk now with precision medicine. Again, just to bring everybody up, to speed in terms of where we are going with this. Precision medicine is I say it's a new vogue or a new thing, but it's really been around. If you look at something like blood transfusion, we've been doing precision medicine for a very, very long time. But what's the difference now? I think the difference now is that we have access to genomic information in a way that we didn't before. And we are actually now trying to say, how do we use all this information to better target the advice we give to individuals and the way we treat individuals? But perhaps for me, what genomics is really helping us to do is to understand biology in a completely different context. For example, we used to treat cancer based on organ-specific therapy, but we now know that some genetic alterations are common to multiple cancers and therefore may require the same therapy. Okay, so I believe that if we do this correctly, that we can begin, again, this is the same APOL1 story, we can begin to give very good public health advice that people can use, in terms of, for example, in the APOL1 contest, you want to be careful before you donate a kidney if you carry 2 copies of this variant, because the likelihood of the other one failing is quite high. Okay? So, and if you're also a kidney recipient, you may also want to be a little bit careful where you are receiving your kidney from, because if your donor has 2 copies of these variants, the likelihood of your kidney that you are receiving to fail is also quite high. So again, you can see how this can give us some, direct public health implications and clinical implications. So we all know that NIH has launched the All of Us program with funding from the White House during the Obama era, and this is ongoing work now. But the point I want to make here by showing these beautiful colors and ethnic background is that maybe for the very first time, precision medicine may actually move us beyond the use of race in clinical practice. Because right now, if I walk into a doctor's office, they're going to look at me, okay, this is African, this is that, this is. But they're forgetting that if you look at me and my mother, we may carry very different variants for a specific condition. So by looking at me and making assumptions about me, you may indeed be putting me at increased risk or giving me an ineffective drug. So we need to maybe at some point get to a point where we are actually basing our decisions on individual genotype, not necessarily the fact that what we see phenotypically. So that brings us to the era of genetic risk score. One of the challenges that GWAS findings has faced over the years is public health individuals and epidemiologists and just generally clinicians have been saying, yes, GWAS is good, but what has it done for me lately in terms of improving the health of my patients? So this is the very first attempt that I think of putting together how we can put together the various informations that we get from GWASs and put that in the context of trying to understand public health and how we can advise people. So genetic risk score is basically taking all or just about all of the information we've gotten that are significant in GWASs and try to use that to stratify. You compute a score and use that to stratify populations, but it needed to be tested to see if it's actually quite effective in stratifying people. So it turns out that if you do it very well using very sophisticated methods, as you can read more about it in this paper, that indeed you can indeed stratify people at maybe greater than 3-fold risk. You can cut that, increase those folds as much as you can, but it turns out that about 8% of individuals who carry high-risk variants for coronary artery disease can be identified. But what is the importance of this? You do see that if you treat these individuals maybe early with statin, you can indeed alter their risk of coronary heart disease. To me, that's really the message here, is if this stands, that we can actually use risk profile from GWAS findings to stratify people such that we can start early intervention for something like statin or even lifestyle changes, then we can begin to see how the interface of genomic findings and public health can indeed be put into practical use. But there are a lot of ethical issues here that needs to be worked out. For example, one of the papers that I reviewed recently for a journal, I asked the question, what is the stability of genetic risk score over a life course? Okay, yes, if you screen a child at birth, because I think we are going to get there, sooner or later all of us, we are going to carry our genome as our driver's license. And we're going to sequence people when they are born, and we're going to have all of that genetic information. If we compute a genetic risk score at birth, how good is that information at age 40? How predictive it is at age 60? I think it's all some of the things that we need to test. and understand? And also, are we giving parents too much power over the life of a child by giving the parents this kind of very strong genetic information of birth? How will parents use it in terms of bringing up their children? If a parent knows that if a child is having a variant that's maybe increasing their risk for coronary artery disease, will they stop them from exercising, from playing sports, from doing all of those things? Are very strong ethical issues that we don't know how they're going to play out with time. So we just need to study them more. And I thank you. And that's the dean made some remarks about that. This is one of the most rewarding awards I've ever received. And I'm extremely proud to be back to the School of Public Health here and to see it growing with a new dean inviting me here to give this talk. I really, to me, I want to show my real appreciation and thanks, for that.